Okay, welcome. I'm going to go ahead and get us started. I'm Kate Cagney. I direct the Institute for Social Research, and I'm delighted to welcome you to this session of the ISR Insight Speaker Series. Our speaker series is our way of opening the door to ISR, sharing the innovative work of our faculty, and to understand and improve the world around us, and to get guidance from our key faculty how to do so. With over 300 faculty, 1,200 staff, ISR research spans an incredible range of topics from wealth inequality, racial justice, to political opinions, aging, and health. You can find the full schedule of upcoming talks on the website, isrumich.edu. And just a housekeeping note, today's talk is being recorded, will be posted there as well, so you can revisit it and share it with your colleagues. And it is my great pleasure to introduce today's speaker, Grace Noppert. Again, so Grace is a research assistant professor at ISR Survey Research Center and a faculty associate in the Population Studies Center. Her work sits at the intersection of biology, sociology, epidemiology, and examines how social experiences across the life course, such as socioeconomic status, shape our biology. She's also quite interested in research on rural life, and we're discussing collaborations. I will say that Grace wins the award for most provocative title I think we've ever had in ISR's 75-year history. Let's talk about herpes, what chronic viruses reveal about our lives. So clearly she's discussing herpes, also CMV, and sort of how these act as biological archives of our lived experiences. So I'm really interested in this notion and how that plays out theoretically and empirically. She's drawing on population research and social infectious disease, and epidemiology more broadly, immunoepidemiology as she describes it. Grace will show how persistent viruses reveal the connections between early life environments, social inequality, immune aging, and I'll just say right now, the Longitudinal Studies of Aging meeting is happening upstairs. Grace will participate in that later, but this was a key theme that emerged from our morning discussion. So they'll look forward to seeing you and your guidance from today's talk. So we'll have time for Q&A at the end of the presentation. Please enjoy. Oh, and there's something, this is interesting. I'm not sure what this means, but Grace will let us know soon. So with that, please welcome Grace Noppert. Thank you. Thank you. Thank you so much.
Can you guys hear me okay? So thank you to Kate for the invitation to present today, and to Catherine. And actually, this talk is because of Catherine. We were working on something together, and she said, I've never heard somebody talk so passionately about herpes. I was like, well, this is what I love to talk about, so I was really excited to get to do this. So as way of agenda, so we'll first talk about what are herpes viruses, and I will tell you why you received that letter when you came in. And then we'll talk about the connection with health, and then we'll end by talking about herpes in our social worlds. Because there's a reason why this work is perfectly situated within ISR and what we do here at ISR. And also that's the part I most love to talk about. So the first thing, stand up if you're able, if you were given the letter B when you came in. Okay, now look around. So statistically speaking, the people with the letter B are the only ones that don't have a herpes virus in the room. So I'm sorry to say the rest of you probably do. And you can sit back down. And I went easy on you guys as far as population prevalences, because we really don't know the true prevalence of any herpes virus in the population. So I went with 70%, but it's probably closer to 90%. So to give you a background on herpes viruses, these are some of our longest viral companions, and our genome is made up of a ton of different viruses. So these are part of our everyday worlds. But herpes viruses are particularly one of the most ancient companions we have, and they infect nearly every person worldwide. And what makes them really interesting is that they establish permanent residence in our body. And I'll talk a lot about what that means for how we understand health and aging later. So about a 90% global infection rate, so I really did go easy on you. There's 8 human herpes viruses. I'm going to focus on 5 that we measure more regularly, but there's 8 that can infect us. And these are the ones that I'm most focused on in my work. And I wanted to give you a sense of, these are ones I'm sure you've probably heard of. So herpes simplex 1, which is what causes cold sores. And you can see the way that it spreads and kind of the estimated seroprevalence in the US, and then the primary age of infection. I do want to note here, repeat infections happen across your life course. So this is probably when you maybe first got it, but not the last time you had it. You have HSV2, which is genital herpes. When I give these talks, I really don't like people to tell me their clinical stories about herpes because I'm not a clinician, so please don't. There's varicella zoster, which is chickenpox or shingles. You know, I'm of the generation where we all got chickenpox as kids, right? And I had it like, you know, the marks all over my body. Now we have a vaccine for that, and we have the shingles vaccine. And in a few slides, I'll show you why the shingles vaccine is so powerful. There's Epstein-Barr, which almost everybody has. And then some people clinically present with mono. A lot of high schoolers get mono, right? College kids get mono. And then we have cytomegalovirus, which is really studied in the context of aging a lot. And we have a lot of our population studies on aging are collecting data on CMV now. This is the one that I would say is more quietly doing its harm to you, right? So with CMV, something like that, you don't usually ever have clinical symptoms that you go to the doctor for. You feel kind of crummy, but you're like, maybe I have a bad cold. But then what happens over the rest of your life course is what's really important. I think about if you have kids, right? Or if you have kids in daycare or you have kids in school, you almost certainly have one, probably more than this, right? Kids are bringing home everything. If you were ever a school teacher, which I was, I'm sure I have probably all 5 of these. So anyway, there's just to give you a sense of these are really ubiquitous in the population. And as of yesterday, it's not just a problem in humans. My friend John Kubale over in ICPSR sent me this headline about horse herpes virus outbreaks in Michigan and the problem that is for the horse population here. And newsflash here, they got it in Texas and they brought it back. But just to give you a sense of these herpes viruses are everywhere. So focusing in on CMV a little bit to give you a sense of the numbers here, right? So 1 in 3 kids are estimated in the US to have CMV by age 5. 50% of adults are positive by age 40, and then 65% of adults age 65 plus are positive. I got that last piece of data from the amazing work that the Health and Retirement Study here at ISR has done of collecting, and they were one of the first population studies to do this, to collect data on CMV so that we can start looking at how it's operating in the body. So how do they work in the body? Okay, so there's this really interesting lifelong tug of war that happens with herpes viruses, and we're just trying to figure out how do we actually measure this whole process. But here's what we think is happening. So sometime early in the life course, say daycare, preschool, school, you get a primary infection. You get your first infection with something like CMV. And then what happens is your immune system puts all of these resources into getting that virus down into latency. What that means is you got to stop the virus from replicating. When the virus is replicating, it's doing its harm. When the virus is not replicating, it's kind of in a sleeping state. You don't ever clear a herpes virus, but you can get it into this dormant state. And then something happens and it triggers it and it reactivates and this whole process starts again. And so we think this process is happening over and over and over again in the life course. But each time this happens, it's costing your immune system something. So what is it costing your immune system? So in your immune system, so the other thing I love to think about are T cells. So interesting. So in your immune system, you have T cells are one of your main immune cells, right? And what you want is you want these young svelte T cells, and we call those naive cells, meaning they're not differentiated for any particular virus or bacteria. They're ready to take on the world. And so the more undifferentiated they are, the younger, the healthier, the less inflammatory they are, the better they're at as functioning. And so when you have a herpes virus reactivate, what it does is that some of those young T cells have to differentiate to be specific to that herpes virus. So all of a sudden your pool looks, okay, has a little bit more of the memory cells. No big deal, we go on. But suppose that process happens again and again. And all of a sudden, your pool of T cells looks a lot more aged and a lot less functional. And then that's causing your immune system to accelerate it's aging at an accelerated pace. What that means is those memory cells, and memory cell T cells are incredible, but they are surveilling your body at all times to look for the virus or the antigen that's specific to them. But if you don't have any naive cells, what happens when you have a brand new virus or a brand new bacteria and you don't have any naive cells to deal with it? to start differentiating to make the antibodies that you need for it. So that's why we see among older populations, vaccines aren't quite as effective. They don't have as many naive cells to deal with that, mostly because their pools are really differentiated for herpes viruses. So herpes viruses tend to take up a lot of space in the immune system as we age. So how do we measure the impact of this process? So usually higher titers to herpes viruses, that's what we're looking for. Higher titers in this context means your immune system is less able to deal with these viruses. But I do want to say higher titers is usually a good thing, right? I get my titers checked pretty regularly for like measles, mumps, and rubella to make sure that if we were to have a measles outbreak, I'm good to go. You usually want higher titers. In the context of a persistent virus like herpes, higher titers is an indicator, kind of indirectly, that your immune system is accelerating with its aging process. You also look for a higher proportion of T cells specific to a herpes virus like CMV, and both of these are indicating advanced immune aging. So if we really zoom in on the CMV paradox here, so it's this chronic activation that is causing the exhaustion of the immune system, and it's causing the immune system to be in this kind of state of constant vigilance. That pressure is accelerating the process of aging, and it reduces our ability to respond well to vaccines, and it reduces our overall immune competence. And I think the immune system is the most important system we have in the body, and it's leading to all kinds of other chronic conditions, including higher rates of morbidity and mortality among older populations. We know now that there's a lot of building evidence here that herpes viruses are risk factors for a lot of age-related diseases. It used to be that nobody paid attention to these. They thought they were nuisance viruses that we could just kind of ignore. And now we know that herpes virus load is connected to cognitive decline, which I'll show you some more evidence on that in a second, that it's part of the etiologic risk factors for cardiovascular risk. And then this also opens up a lot of opportunities for therapeutic interventions, thinking about how do we get better vaccines for many herpes viruses, not just varicella zoster. I want to highlight two recent studies that have been really groundbreaking in this area. One is looked at a population of older adults who got herpes zoster vaccination, so shingles vaccines, and they saw a reduced prevalence of dementia in that population compared to those that didn't get the vaccine, because that vaccine is helping your body deal with that herpes virus. The other one was a very elegant epidemiologic study that was done a few years ago, this longitudinal analysis of I think it was people in the armed forces, but they looked at the prevalence of Epstein-Barr and its association with multiple sclerosis and found that Epstein-Barr was causally linked to MS. Now, that doesn't mean that everybody that has Epstein-Barr is going to get MS. It just means that in the constellation of risk factors for MS, Epstein-Barr is one of the ones that has to be in there, which is quite astounding. This little virus that we don't think is causing many problems is actually causally linked to multiple sclerosis. In our own work, done with a lot of folks here at ISR, Ken Langa and Lindsay Kobayashi, among others, we see this is looking at domain-specific cognition. So can we see an association between your levels of CMV IgG, so higher is worse, and different domains of cognitive function? And this work it's under review right now, but what we see is that we see these robust associations between CMV and executive function in particular. So it seems like there might be some very specific domains involved, but we're not sure. Does that mean the virus is entering the blood-brain barrier? Does it mean it's causing peripheral inflammation, which is causing neural inflammation? This is all kind of brand new work. So it's an exciting time to be in this field. We also know there have been studies a few years ago looking at CMV and its impact on mortality, and we know that those that are in the highest quartile of CMV response, so they have the highest titers, their immune system is struggling the most to keep CMV in that latent state, they have the worst probability of survival over time, and that's this line down here. Okay, so now I gave you like the why we should care about CMV for the health part, but now for the really fascinating part, which is the social part, which is why this work should be done here at ISR and why it's really fun to get to do it here. So herpes viruses also act as an archive of our lived experience, and that's what I'm going to try to prove to you over the next few parts. So I want to start with this really great figure from Kathy Harris and Tom McDade from a few years ago, because I think this helps sets the stage for how we understand this process. So what they do here is they lay out that there's two big domains. There's the social domain on the top with its different hierarchies from interpersonal relationships all the way up to neighborhoods and social structures and political economy. And then there's that analog system for the body down below. So biochemical messengers, which is often what we're thinking about when we say biomarkers, organs and tissues, physiological and neurological systems, and the genome and the epigenome. What they lay out is that those two systems are interacting with each other across your whole life course. So they're inseparable. And if you want to understand health, then you have to start understanding the intersection of the social and the biological. You can't do this biological work without understanding the social environment and vice versa. And especially if you're thinking about aging work, you can't just, when you see these health outcomes in older adulthood, you have to start trying to understand the story that was being told across the life course about this intersection. Well, let's see. Okay, so let's go back to this figure, because I think this figure can also help us understand how social experiences become biologically embedded through the lens of herpes viruses. So we've got this system, right? This reactivation and latency, et cetera. But what causes these cycles? And we think that when you have a stressor, that it causes your virus to reactivate, and this whole thing happens again. So when I give this lecture to students, I tell them, like, how many of you guys remember, and you guys might remember this too. It's been a while ago for me, but when you would take finals in college, and then you would go home for Christmas break, and you get super sick, right? Like, I see a lot of head nodding. I'm sure parents understand that process too. My mom would always be like, why do you come home and you're so sick? Because your body is doing you a solid. When you're in finals, it's pouring all these resources to keep you really healthy at that moment, but it's also experiencing this big stressor. And as soon as that stressor is removed, all of the viruses that your body has been pushing down for you and really helping you out now have the space to reactivate. Right? And you can see that happening with other stressors, right, in your life. Or when you get really sick in a biological stressor. We saw it a lot with COVID. People would get COVID, and then they would have a reactivation of a latent virus because they had a massive biological stressor, and then that virus reactivates, and then your immune system has to do all this work to get that virus back into latency. So understanding the stressor process might be the way that we can understand how these viruses are becoming biologically embedded. So what we think is that herpes seem to be sensing and responding to your social environment and through this mechanism of stress. We think there's this dynamic interaction between your stress response and herpes virus, such that one of the ways that we understand the stress of what's happening in your social environment is by looking at your herpes virus load. So how does the social environment shape how our bodies deals with herpes? So I think what my body of work here is across the life course, our herpes virus loads can reflect exposures at multiple levels. So we can think about the individual experience, but we can also think about family and family socioeconomic environments. And then we can also, which I love to think about, our neighborhood and community context. So we're gonna go through each of those levels, but we're gonna start with the individual experiences. So here at ISR, we conducted a series of experiments or studies, not in the bench here, examining whether reporting having experienced trauma in early life was associated with your antibody levels of CMV in later life. So I want to shout out the Health and Retirement Study first, because they have collected this massive amount of amazing social data, specifically Jackie, to understand what people's life experiences were. And so you have these people that are in their 60s and older, and they're asking them retrospectively, did this happen to you before the age of 18? Did this happen to you before the age of 16? That's the format. And so the first thing we did is we looked at reports of people that experienced parental death before the age of 16 or parental separation. And the first thing we did is said, okay, well, let's first look at their average IgG, which is their immunoglobulin antibody level to CMV. So higher is worse. And we first wanted to say, okay, do we see a difference in their average? And in fact, we do, right? Those that experienced parental death had a much higher level of CMV IgG than those that did not. And the same held true for those that experienced parental separation. But the question is, do these simple averages tell us all we need to know? And the answer is no. There's many life experiences that overlap here, right? So there's a lot of things that could contribute. So to understand whether trauma itself is contributing to the worst control of CMV, we use a series of statistical models that adjusted for age and sex, in parental education, in adult health conditions, so maybe you have a lot of other adult health conditions that you're dealing with, adult health behaviors. And these models then help us separate the trauma signal from the other pieces of life that travel with it. So that's what I'm showing here. And so we stratified these models by race ethnicity as well, because we were positing that the experience of structural racism also patterns your risk factors for being exposed to parental death or parental separation, but also is making you more vulnerable to immune aging because of the impacts of structural racism and discrimination. And so we separated these out by race ethnicity. So this is the experiencing of parental loss. So this is like, did you have a parent die before the age of 16? And what we see here on the top is that among non-Hispanic Black individuals, experiencing parental death was significantly associated with higher antibodies for CMV in later life. So again, this is an experience before the age of 16 and their antibody response to CMV in later life. And we poked at that by controlling for all these other things, and the association was robust. Right? So what was astounding to me about this paper, and it's been a few years now, is that you see this experience that people were reporting on years and years before showing up in the way that their immune system can deal with a small viral infection. It's just wild. We see even stronger associations when we look at parental separation, which it seems we've poked at this a little bit more. We think parental separation might be a more destabilizing event in some ways. And we see this hold for all race ethnic groups, that experiencing parental separation before the age of 16 is significantly associated with higher CMV antibodies. We are now in the process, with my colleague Kate back here, we're now in the process of saying, okay, well, what about, let's zoom out from parental separation and parental death. Let's look at other traumas. And so now we've created this composite score of all these different traumas that people have reported on in the HRS. Were you ever in trouble with the police? Were you physically abused? Did you ever fire a weapon in combat? Were you the victim of serious physical assault? And we compiled them into a score. And what we found here, and this is work that's in progress, we found that people that reported, and this is comparing it to zero traumas, so people that report zero traumas, people that reported 3 or 4+ traumas had significantly higher levels of CMV. And what we did here is we controlled for, in models 2 and 3, we controlled for whether they also experienced trauma in emerging adulthood and midlife. Because we thought, well, maybe it's because if you experience a lot of trauma in early life, then you experience a lot of trauma in your other life course processes. But in fact, there was this robust association with what's happening in early life in particular. And what we think is that early life trauma is priming your immune system in really particular ways, such that it's less able to deal with these viral infections across the life course. So we're still finishing up work on this paper, but we're kind of excited about the findings that were and what they mean. Okay, so we started at the individual. Now let's zoom out to look at families. So this is the work that I actually did with Bob Hummer, who's in the building today from UNC. This was what got me started in this whole area of research. So we did this paper looking at education. So for those of you who don't know Bob's an expert in education, and he's great. His work is amazing in this topic. But he wanted to know, okay, what do we see? Do we see an educational gradient in CMV response? So again, looking at, CMV IgG, we wanted to know, split into quartiles, we wanted to know, do you see this gradient whereby those with lower education are in higher levels of CMV response? So in this top bucket is your own educational attainment. Again, this is in the HRS. And we see a very clear educational gradient. This is comparing folks to College Plus, so they attain College Plus. Those that had less than a high school education had a much higher odds of being in the next category of higher CMV response than those with a college education or plus. And we controlled for several different things to try to see, are these other variables kind of muddying this association? And these were very robust to a lot of different controls. What is even more interesting though, is panel B is the participants reporting of their parents' education. And so we see this very robust response to if your parents had 8 years or less of education, you're significantly more likely to be in the next higher category of CMV response than if your parents had more than a high school education. And this is robust at controlling for your own educational attainment. So this is something like I think a lot about this, right? You're talking about people in their 60s and 70s and 80s reporting on what their parents' education was 60, 70, 80 years ago. And it's still showing up in the way that their immune system is dealing with this viral infection. And it seems to, I mean, we have poked at this a lot. I mean, Kate and I talk a lot about what can we control for and how is this operating and can we get at the mechanism? And it still shows up. There's something about the ways that the social environment becomes biologically embedded. And it seems to be that the herpes viruses are really a window into seeing that process happen. Okay, but what if we zoom out even further? What if we think about neighborhoods and community contexts? Because our lives unfold within these larger structural contexts. And I want to just take a quick minute to make the case for neighborhoods, because neighborhoods is something we love to talk about here at ISR. Our director is one of the leading thinkers in neighborhood work. So neighborhoods are a part of the constellation of social determinants of health. And we talk about part of the conditions in which we live and we play and we work. And so we think about, okay, how does neighborhood context play into health? And one of the reasons I love thinking about neighborhoods myself is that neighborhoods, you can imagine, are changeable, right? So while we work to address things like structural racism and to get universal access to education, those are really big goals and they're good goals. But in the here and now, we can think about policies that may actually make neighborhoods better and more healthy for people to live in. And we spend a lot of time doing this. So we really wanted to think about, can we bring neighborhood context into this work? And measuring neighborhoods is also really important. And how do we figure that out? So to measure neighborhoods, we need consistent high-resolution tools that describe these environments across the US. And I'm going to unabashedly call out the work of NANDA, which we love to work on, which is the National Neighborhood Data Archive. I've even put a QR code because I feel like it would make our data librarian proud that I included this here. Thank you, Lindsay. Because what we're in the business of doing at NANDA is actually creating measures of the neighborhood context that people can use in studies like the one I'm going to show to understand the ways that your social environment is getting under the skin. And so what we did is for this next study, we used 2 different NANDA variables, neighborhood disadvantage and neighborhood affluence. And we chose these because we think they're relevant to the stress that you might experience in a neighborhood, which might have implications for immune function and viral behavior. So neighborhood disadvantage is an average in a census tract of percent of female-headed households with children, households with public assistance income or food stamps, percent with income below the federal poverty line, and the percent of unemployed. And neighborhood affluence is the average in a census tract of households with incomes greater than $75,000, populations employed in a professional or managerial occupation, or population with a bachelor's degree or higher. So there's something to keep in mind here. So this is the work I'm going to show you is based on the census tract that HRS participants lived in. This doesn't mean that this is what your household is. It means this is the kind of little neighborhood that you live in. This is the average of your neighborhood. So the question we have here is, do we see these neighborhood conditions show up in our bodies? The answer is yes. So what we see here on the left is this is the standardized Z-score for CMV IgG, so antibodies. So higher is worse again. And what we see is living in a more disadvantaged neighborhood shows up in your body as a higher average CMV IgG. And again, you'll see in the models, we poked at this by thinking about all the different controls that we can include here, but we see very robust associations. We see the same with affluence in the opposite direction. So living in a more affluent neighborhood is associated with a reduced IgG level of CMV, so better immune function here. But when I talk about this work, it really still astounds me because we're talking about a really non-specific thing like neighborhood SES showing up in the way that your immune system is dealing with cytomegalovirus or herpes virus. So what do these findings mean? So if we use the model 3 here, a 10% higher neighborhood disadvantage score is associated with a 0.12 standard deviation increase in CMV IgG, or a 50% higher neighborhood disadvantage score is associated with an increase of 0.6 standard deviations. The question I get a lot when I present these findings is, well, what does that mean? Like, is that clinically meaningful? I get that question a lot. The fact is this is all really new areas of research. We don't know what it means yet. What we do know is that the social environment is inextricably tied to what's happening in our bodies. And that we know that there are implications for what that means for health across the life course. So I think this is, if this is provocative to you, I encourage you to get into this area of research because we need to recapitulate these findings. We need to think more about what does it mean for this in a clinical setting. How do we make neighborhoods healthier for people such that they're not experiencing that stress that's causing their immune system to fail them and their herpes viruses to reactivate? This just opens up a whole new world of research. So I'm going to piece this all together now. I'm going to attempt to here. So what did we talk about today? So one, I want you to walk out of here knowing that herpes viruses are universal. You almost certainly have one, but their impact is not. Exposure happens early and widely, but our ability to control the virus is heavily socially patterned, right? Such that it's one of the ways that we see social inequality getting into the skin. The immune system, my favorite system, is an amazing storyteller if we learn how to listen to it. And the reactivation patterns that we see in herpes viruses are direct reflections of lived experiences, including trauma and stress and social inequality. CMV in particular is a really powerful lens for aging, and it captures immune system wear and tear and age-related vulnerability, and it can also capture resilience or strain accumulated over decades. Biology and society are inseparable. To understand health, we have to understand the intersection of both, and we can't forget either of those domains. And the final point here is that embodiment is real and measurable. And what do I mean by embodiment? So I'm going to take a quote from Nancy Krieger, who is one of the thinkers of this idea of embodiment. But the idea here is that bodies tell stories about and cannot be studied divorced from the conditions of our existence, that often but not always match people's stated accounts, that people cannot or will not either tell either because they're unable, forbidden, or choose not to tell. And I think our job as social scientists is to learn how to listen to the stories that people are telling. And one of the ways that we can do that is looking at what's happening in their bodies. So why does this matter for science? And then I'm going to tell you why it matters for society. So for science, thinking about herpes viruses can help us identify early life roots of health disparities. Because while the work that I presented shows you what's happening later in life, I actually have this gut instinct that this is happening really early in the life course and we're just not picking up on it yet. But what if we could? What if we could stop that process early in the life course and we could change the trajectory that people are on for immune aging? Think about what that would mean for health inequalities over the life course. I think that herpes viruses are an early warning sign for immune aging, and they are an intervenable warning sign. I know vaccines are not a popular thing to talk about right now, but what if we could develop vaccines for these herpes viruses that really improved how people aged? They reveal mechanistic clues about immune aging, and they offer a scalable biomarker for biosocial research. And they open up new prevention and intervention pathways. But why do they also matter for society? And I think about this when I talk to my mom and my family about this work, like why should they all care about this? For one, it shows us how social disadvantage leaves biological traces. And it highlights why social policy is actually health policy, why improving the conditions that people live and they work and they play and improving the world around us is actually changing the health of our bodies as well. So social policy is health policy. And I think it reinforces the urgency of addressing inequalities really early in life. We need to start early. But I want to leave you with this. Herpes viruses help us read the stories our bodies carry, but they do more than that. They challenge us to change the conditions that write them. And so the next time you get a cold sore, I want you to think that it's not just a nuisance, it's a tiny little footnote in the biography of your life, and you need to pay attention to what it's saying. That's all I have. Thank you so much for having me. Please get in touch. If you have any questions, happy to take them. I have.
In the context of immunology, I was thinking about Kelly Clarkson's song, "What doesn't kill you makes you stronger."
It's a great song.
Yes, in this context, is that true, or does there's a nuance to it? It depends on the exposure, right?
It's a question that we're actually really debating in our lab group right now. It seems to be, and I didn't show this right now, but it seems to be in the immune system that there is some kind of biological threshold. So what we see, and I didn't show these results, but with some of the other immune measures we used, it seemed like your body could take some trauma, but after a certain point, it really fell off. And so there seems to be like our bodies can take so much and then they can't, and our immune system starts to fail us. With herpes viruses, I think there's part of it is that we can develop resilience, and maybe that means that we don't reactivate our herpes viruses as much, but we don't have the data for that yet. So I think I would love to test that question. And if I did, I would use that song in the title. Thank you for that. I would love to figure that out. That's like the burning question that keeps me up at night. Is like, how do we measure that? No, we're not able to right now. What you would have to do is you would have to be able to test people kind of in quick succession multiple times to see, and you would also have to, and you can do this, but you'd have to be able to detect if they just recently got an infection. So we did this study in a small sample of folks here at Michigan Medicine where we were able to disentangle, did they have a recent or an active infection, or a late infection. Everybody had a late infection. Nobody had been recently activated. So at least we could pull that apart. But you need a longitudinal study that really collects these CMV data at multiple time points in quick succession, at least for like a 6-month period to really get at that cycle. So that's our hypothesis of what's happening. I would really love to be able to prove it. Questions? Dr. Cagney?
That work. I wonder if you wanted to say more about the socioeconomic disadvantage piece, and also, how you made the choice to compare it with an affluent community and what you imagine they both deliver. You alluded to the notion of stress, and I could imagine sort of stories of hypervigilance in the community, and that could be something that could exacerbate. But I wondered if you wanted to.
I think it's a few elements. So I think first of all, there's the concept of social stressors and biological stressors that happen in this case. So like living in a disadvantaged community puts you at much higher risk for pollution, which is going to be a biological stressor. It's a biological immune modulator. So that's going to play in. But I also think there's this piece that is just inseparable in the story of vigilance of you. If you live in a disadvantaged neighborhood, you know that you live in a disadvantaged neighborhood and you have to be hyper aware of that and hyper vigilant. I also think there's the stress of knowing that you don't have access to what you need and you don't have care. I think access to care and delays in care are a big part of that. I also think access to healthy foods and places to exercise. We know that those things reduce stress in your body and are improving immune function. With affluence, when we look at those affluent measures, it really seems like it's getting at something about social capital and power. And I wonder what that means for your immune system, right? Like it could be, there could be a part of it that means, maybe you're more stressed out, but also I think it means that you are more comfortable in the spaces that you live and you feel like you have more agency. And I think that might get under the skin too. But it would be fun to kind of
draw up that side of the distribution.
Yeah, to tease that out. Yeah. That's a good question. Good neighborhood question.
May I just follow up that question on neighborhood?
Yeah.
So I would hypothesize that people in my parents' generation during their experienced when they grew up, they had less information about what's going on in other communities than my generation and then my kid generation. So to the extent that oh geez, people are doing much better than I do.
Yeah.
Or to the extent that better that, oh geez, I'm much better than other people.
Yeah.
Right. So I would hypothesize that kind of the impact will be moderated by the availability of information about other neighborhoods. But I don't know whether we have information to test this.
So it's the social comparisons hypothesis, right? And I think that's a really interesting question. I would have to think about. I'm sure that we could figure out a way to test that, especially in the HRS. But this idea that if all the neighborhoods around you are not doing great, is that less detrimental for you than if you're the sole island of not doing great around a really affluent community? Am I getting at your question? Yeah, I love that question. I have not thought about that yet, but it's a really interesting one. And we know that there's a lot of work in the social sciences about the detrimental effects of comparisons. Like social comparisons are kind of the worst thing for health. So I would stand to reason that it would stand for neighborhoods as well.
I'm also wondering if you thought about urban form and how this is sort of playing a role in these interactions. I'm especially thinking that there's a large difference between coming from a suburban, spread out community that sort of decreases the opportunity for positive social interaction versus living in a dense urban walkable neighborhood. And I think that could also potentially explain why you might expect wealthy people, even as a group, they might live in communities that, despite just this aspect of wealth, they also live in potentially more socially connected. And so I'm wondering if you have any evidence on that.
I love that question too. It gets a little bit at the work that Kate and I have been talking about, rural communities too, of like what social connection look like there and what does that mean for health. We have a study underway right now looking at social connection and immune function to try to start teasing that out. I haven't looked at that in the context of the neighborhood work, but it's a good idea because basically what you're saying is, are these affluent neighborhoods also more socially connected, and so that could even, and you could contrast that with an affluent neighborhood that maybe wasn't socially connected, and yeah, I love that idea. I haven't done it yet, but it's on the docket.
I'm just wondering about the potential to look at clustering, either clustering right within neighborhoods or clustering within families to see if some of these are shared experiences like parental separation, and whether you would see that heightened effect, right? And also imagining from the social disadvantage piece, would you sort of see this at the neighborhood?
One of the things I think about a lot, and we get this critique on our trauma work, is that trauma is really differentially experienced. And how do you capture that? And is that part of what we see in the immune system? Is that, if my sister and I go through the same traumatic experience, the way that becomes embodied for her may be very different than the way it becomes embodied for me. I'm not sure, other than having a study that has siblings, that you could tease that out. Yeah, I'm gonna get them to collect a lot of biomarkers for me. Yeah, I love that. Is Tom in the room? Yep, here we go. But it's an interesting idea. I think you could also, look at, is this what you're getting at? Like you could look at clustering within neighborhoods too and see if that.
I'm also wondering about potential reinforcing that. There's a way in which it would even heighten the experience. Because those surrounding you are
right. One of our analysts in SCH, when we were doing the initial work on parental loss and separation, had this idea in HRS to look at, what if you lost a parent, but you were in a really connected neighborhood? Does that change the way it becomes embodied? And I think it's such a great question. Like, if you can capture, you have some of these communities that may be disadvantaged, but also tight-knit and kind of come around people when they experience a trauma. That's got to be very different than when you experience a trauma and you're extremely isolated and you have access to no one. So I think it would be a really interesting way to push those findings even further. Okay, we're all good. Thanks, guys.